Hey, all right, everyone. So this is our final keynote of the event. I kind of realized it was accidental that yesterday we had, the whole point of the keynotes is to expose data people to fields that they might not necessarily be in. Obviously, there's going to be some people from every field at a conference like this, which is our goal. But on the first day, we kind of had a couple talks exposing people to deep questions about ethics of data, making us confront uncomfortable questions that we probably need to confront more often but don't. And today, the theme, accidentally, is exposing people to the use of data in science and statistics. And now we have an amazing speaker from the land of neuroscience, injecting the knowledge into all of your brains about brains. So Jeremy Freeman is a researcher from one of the coolest scientific places going on in the United States right now that is called the Janelia Research Campus. And it's basically the Bell Labs of neuroscience. So they give people a lot of freedom to discover basic science about how the brain works. And what I really admire about Jeremy's work is that while a lot of the other labs in the facility, this is a good thing too, build new kinds of microscopes, which is a very hard problem. Jeremy interprets the understanding of science as investing in open source to basically write better software for all of science. So there's not a lot of scientific initiative to write good software because of a lot of the broken funding models for how people get paid to do science, and a lot of science is dependent on publishing in journals. But I think Jeremy is a leading light in the world of making scientists encourage and incentivize better software and open source adoption. So with that, I'll let Jeremy get into it. Thanks.
Awesome. Thanks so much, Max. You basically just gave my talk. So what he said. Thanks so much for having me here. And it's just been an amazing conference. The diversity of topics that I've been exposed to has really been remarkable. So this is an awesome conference, and there should be more like it. I'm Jeremy, as Max said. These are some of the ways to reach me on the internet. And I'm going to be talking about open source neuroscience, which is the sort of broadest phrase I could come up with for describing the variety of things that we do. So this is Janelia, the place that Max was talking about. It basically looks like a spaceship that landed in the middle of the forest. It's in Northern Virginia, and there's not a lot going on in Northern Virginia, but there's this place. And it's a super cool research facility. It's nonprofit funded by the Howard Hughes Medical Institute. And as Max said, a lot of the focus at Janelia is on neuroscience, and it's on the development of technologies for neuroscience. So there's a lot of work on genetic engineering methods for designing animals with neurons that express cool proteins that do interesting things. And there's also a lot of work on microscopy methods, so new ways of looking at the brain and interacting with the brain. But then my group is mainly computational, and we work collaboratively with a lot of the other groups there to basically understand the brain and try to develop lots of open source tools for helping in that effort, both at Janelia and also in the broader neuroscience community. I've really valued at Janelia that they support that. I've really appreciated their investment and support of open source, and that's meant a lot to me being there. It was actually Max that pointed out to me that Janelia looks a lot like the Starfleet Academy. It's not, unfortunately, next to San Francisco. Well, for better or for worse. But it looks a lot like that, and kind of feels like that while you're there. So I'm gonna start this talk by telling you sort of the motivation or some of the reasons that we study the brain, and a little bit about what we know about the brain, also what we don't know about the brain. And this was really brought home to me pretty recently when I gave a talk at a library to a bunch of fifth graders. If you ever want a really humbling experience, you should talk to fifth graders. So I was supposed to be there to tell them all the cool things we know about the brain. This was the local Loudoun County or in Ashburn Public Library. And I was starting to tell them all these things, and they started to ask me these really hard questions. And they basically just started interrupting me. And they said, which you should all feel free to do as well. And they started saying things like, this one kid said, how is it that I can hear a phone number, and then the next day I still remember that phone number? And I didn't know how to answer him. That's a really hard question. And this other kid was like, why do I always dream about robots and dinosaurs? I was like, I don't know, that sounds really cool. I wish I was you. So the funny thing about the brain is that even over the last 50 years or 100 years or 150 years, we have developed enormous capabilities in terms of our ability to measure the brain and to manipulate the activity of neurons in a wide variety of animals, less so in humans, but especially in animals like flies and zebrafish and mice. But conceptually, there's still an enormous amount that we do not understand. I wasn't able to answer those fifth graders. I probably can't answer any questions, well, hopefully some, that if you were to ask me right now. And that's because there's just still a lot we don't understand. So a lot of the collaborations I'm involved in are trying to do the best we can to understand the brain by leveraging all of these cool new technologies that we have. And what a lot of that looks like, and I'll just give you a brief sort of overview of the kinds of experiments we do. That looks like taking animals. We do a lot of work in the mouse. The mouse turns out to be a cool model system in terms of both the capabilities we have for measuring and manipulating neural activity, but also the behavioral complexity of the animal. So we do a lot of work in the mouse. We often put mice into virtual environments, and I'll show what that means in 1 second. And while the mice are in these complex virtual environments, we record neural activity using usually microscopy methods, in particular 2-photon imaging. So the way this works is the animals, the mice, are basically genetically engineered so their neurons express proteins that literally light up when those neurons are active via calcium binding. And then we measure that in the form of images using 2-photon imaging, and I'll show what that looks like in a second. We also make electrical recordings from the brain using electrodes. So mouse VR looks something like this. And when you hear VR, you might think about visual VR, which for humans makes a ton of sense. Mice do have a visual system, they have eyes, but it turns out that they experience the world much more predominantly through their whiskers. So that's a pretty hard thing for, I think, a lot of us to understand. I don't have whiskers, so I don't know what that's like. Maybe Max can tell us about the experience. It turns out to be a really important way to get mice to experience the world. And this is the setup built by an incredibly talented former grad student, now postdoc at Janelia, working with Carl Sabota as well as me, Nick Safroniev. So Nick's Tactile Vr System, the mouse is on this ball, and there are physical walls on either side of the mouse moving back and forth on little motors. And as the mouse runs on the ball, in one way or the other, the walls move back and forth in closed loop with her running on the ball. And that gives her the experience, based on sensing the walls with her whiskers, of being inside this virtual environment and basically running through these winding corridors. So this is a very compelling sort of illusion or sort of virtual experience for the animal. You basically drop them in and they start doing this right away. So this is a great way to now start asking questions about what the brain is doing while this animal is exploring this virtual environment. And this is a picture of Nick at the rig, just to give you a sense of what this sort of looks like on the ground. He's faced with this incredibly complex collection of user interface components, which we're trying to rewrite in JavaScript. That's another weird project that I might talk about later. Here's Nick actually looking at some data where he's recording brain activity in the form of images and looking at them in a Jupyter notebook during the experiment and doing some data analysis basically online. A lot of what we want to do in these experiments is analyze the data as quick as possible, because that lets us make decisions about basically what experiment to do next. So those data that was just a glimpse of look something like this. This is actually from a very recently developed microscope at Janelia that lets us look at an even larger portion of the brain than we were really able to before. So what we're seeing here in this picture of flashing lights, each one of these tiny little spots that you see that look like little stars are individual neurons. And we're looking at the activity of neurons over a really large fraction of the brain. This is part of the brain involved in processing somatosensory information, so information from things like the whiskers, as well as other areas. And this is using a method or a new microscope called the random access 2-photon mesoscope, developed by Nick and also Dan Flickinger and Carl at Janelia. And while the mouse is in this complex environment, we can look at these really interesting spatiotemporally rich dynamic patterns of brain activity and then try to understand the relationship between the world that the mouse is experiencing and what the brain is doing, and how the brain might be responsible for or enable the mouse to do all the rich, interesting, complicated things that it can. To give you some perspective, even about maybe a year ago, or yeah, exactly a year ago, the microscopes we were using let us basically look at something about that size on this picture. So this is an incredibly exciting development, a microscopy development enabled by collaboration between these people at Janelia. So we can look at a lot of really cool things in neuroscience right now, and we're basically on the verge of, yeah, question?
Just to understand the image, it's just the surface, right? It doesn't have the ability to look sort of volumetrically into the...
No, it does have the ability to look volumetrically, yeah. So this is layer 2-3 of the mouse cortex, and we can image with this microscope at a variety of different layers. The cortex is organized into layers. And we can look at layer 2, 3. We can look to some degree at layer 4. And yeah, actually have a surprising degree of access, both there are trade-offs in space and time in terms of how fast we can image and what volume we can look over. But we have pretty significant access. We can basically, with this microscope, image or look at activity across what in your brain would be like that. Of course, we can't do it in your brain. Not yet. So we have these amazing tools. We have these amazing measurements that we can make. I spend a lot of my time thinking about what I call the 80-20 problem, based on some things that people told me recently about labs, which to me is really the problem of the amount of time that we spend making these incredible measurements and the amount of time we spend doing everything else. So I was recently talking to some labs who basically described this phenomenon where it used to be the lab would spend 80% of their day, or the students in the lab would spend 80% of their day making measurements and doing science, and 20% of the day would be spent, moving the data around and doing a little bit of data analysis. And that now things have basically totally shifted. So labs and every single grad student will spend 80% of their day just kind of mucking around with data, and not even the fun, interesting parts, just like moving data from one place to another, and like relabeling things and trying to organize things in a sort of haphazard way. And then they spend 20% of their time actually doing science. And this is something that really struck me, especially for some of the kinds of experiments that I was just showing when I got to Janelia, and I saw this all over the place. And I've been trying to spend a lot of time understanding, first of all, like why we're in this situation, which has in some ways become like an ethnography of labs, and trying to understand why labs are in this situation, and then how we can maybe try to fix it. So I think a lot of this happens because when we think about data analysis, we sort of have in mind a picture that's going to look like this, where you're going to have data, and then you're going to do something, and it's going to be really easy, and then you're going to have some cool result, and you're going to publish it in some cool journal. But in practice, this is not what happens, because it looks a lot more like this. And I'm sure everyone here can sympathize with this experience, but I think it's something that often catches people off guard. And that when you're starting in a lab or you're starting to do an experiment, you sort of think, oh, I'll just build my pipeline and I'll get from point A to point B, and it's going to be really straightforward. But then you end up in this situation, and I think there's a few reasons this happens. One of them is because the data are a little bit large. So I don't think the data we deal with in these experiments is quote-unquote big data. It's actually not that big, because maybe a given imaging experiment is 50 gigabytes, at most maybe a terabyte. So it's nowhere near, I don't know, like what the Large Hadron Collider deals with, but it's also sort of big enough to be annoying data. And that I think is a huge problem, because a lot of this mess and complexity comes from just the challenge of sort of moving stuff around and dealing with stuff when you're not really using the right tools and things get big enough to be kind of difficult. The other big thing is that there's an enormous amount of sort of reinvention across the entire scientific community. And this was really I was struck by this when I started to sort of research how different labs solve these problems. And what I found was that almost every lab was basically completely independently reinventing a pipeline to solve exactly the same analysis problem. And it was usually some complicated combination of scripts that weren't really versioned in any particular way. And they sort of got the job done, but it meant that there was no sharing of infrastructure or tools or standardization of tools across labs, and which obviously has a lot of drawbacks. We spend a lot of time reinventing the wheel. We don't end up with things that are stable and sort of work across a lot of different systems. So the picture that I want to turn this into, and this is honestly really inspired by Max and by a lot of people in the Node community who taught me about hexagons, is really to try to turn it into a picture like this. And I both love this visual metaphor, and I really think this is what we want. I think we need to think about these pipelines that people are building not as monolithic pipelines, but as basically collections of modules that solve really well-defined small problems. And some of those are going to be super general. Like when you're working with this kind of imaging data, literally everybody has to solve some core set of problems. Everyone is registering their images. Everyone is finding groups of pixels that correspond to individual neurons. Like literally everyone doing this work is solving that problem. There are a lot of problems that individual labs have to solve and other labs don't. So there are going to be parts of this collection of modules that are specific to some labs, and there are going to be parts that are really general. But if we start by thinking about these, as opposed to starting by thinking about building the monolithic pipeline, then we can build pipelines out of these modules and work more collaboratively across groups to try to solve these problems. So this is, I think, a vision for what, as a community, we want to achieve. And in my group, we've been trying to do some open source development to build some components of this. First of all, I don't think there's any particular language or framework or toolkit that's going to be the only thing we use to solve this problem. So in my group, this is a variety of the kinds of things that we use. We do a lot with Node and WebGL, well, for a variety of things, but a lot for data visualization. We use a lot of cloud web services, which I think are way, especially in science, way better than relying on institutional clusters. I can talk more about that if people are interested. But I think an over-reliance on local, centralized, institutional infrastructure for computing has led to a lot of issues with sort of transferring work from one group to another. We do a lot with Spark because, as I said, so Spark is a distributed computing engine. Again, these data are sort of big enough to be annoying, and that means you want something that can help you with that. We also do a lot with the Jupyter Notebook, and I'll say more about that later. One thing that's not on this list, and very deliberately not on this list, is MATLAB. And I don't know how many people in this community have dealt with the perils of MATLAB, but it's a proprietary sort of computing platform that happens to be what about 99% of neuroscientists still use for most of their data analysis. It's closed source. It has a number of problems for doing, I think, really any kind of effective software development, and it's not open. So that's not on this slide. We also have been working on the two projects in the lower left, Thunder and Bold. And these are two open source projects that Jason Wittenbach in my group has been really involved in developing, and links to them are here. And just to say very briefly what these are about, it's really about solving a lot of different related problems in image and time series analysis, which turns out to be exactly what we're doing most of the time with these data. These large images of neurons flashing over time are essentially collections of images. You can also think of them as time series. And there's a lot of analyses that we need to do with these data. And the goal of Thunder is to basically build a collection of modules that solve different parts of that problem. So some of those modules, this is all written in Python, some of these modules are related to just efficient storage and manipulation of image and time series data, which is kind of cool because it turns out people have now started using this in a lot of domains way outside of neuroscience, which I think makes sense because there's a lot of commonality. But then other modules as part of this are really for specific problems like image registration or tracking neurons or segmenting neurons and that kind of stuff. And the goal is to have, again, a collection of modules, some of which are really specific and some of which are really general, that we can use collectively to solve these problems. We've also spent some time recently thinking about standardization, not so much at the sort of code level or modularization at the code level, but in terms of the algorithms we use and the data sets we're running them on, and how we think about vetting and standardizing those algorithms across labs. So one of the really amazing things is that basically every single lab doing this kind of work, and there are hundreds, are solving at some point the exact same algorithmic problem, which is really kind of simple. It basically involves taking images like this one and identifying groups of pixels that correspond to individual neurons. So I said those flashing lights are neurons, but you don't want to just analyze a movie. You want to talk about individual neurons, so you have to find which pixels are neurons. This turns out to be a sort of weirdly hard problem. If you think it's trivial and you think you know how to solve it, that's great. I'll tell you what to do in a second. But it turns out to actually be pretty hard, and it's something that, again, every single lab doing this work needs to solve. But instead of having some kind of shared standardized infrastructure or even just shared anything for how to do this, basically every lab has come up with their own independent way of doing it. So the goal of this project, NeuroFinder, is to basically try to bring some standardization to this problem. So what we did, and we just put this up really recently, we put together a bunch of data sets contributed from a bunch of labs that we got to basically just put their data in a standard format and give us some kind of ground truth. Getting ground truth is complicated, but just assume that we did it. And we have training data sets, we have testing data sets, and now people can basically submit algorithms by just going to this website and putting their information in and dropping the result file that tells us on the test data what they identified the neurons to be. So far we have only basically just been testing it with our own submissions. These were mine. One is called random algorithm and one is called terrible algorithm. This was just to make sure the system works, though it's not that bad. It's only 10%. We just recently launched it, and we hope that this is an example of, question?
Why did you use this system instead of, for example, a Kaggle?
Yeah, so we definitely looked at Kaggle. There were a bunch of things that we sort of felt we needed that were kind of custom to this setup. That was originally why we just felt, I don't know, it seemed like a simple enough problem that building something that we had sort of some control over and then could reuse for other problems in our space, as opposed to necessarily buying into that particular ecosystem. Seemed pretty easy. That was the main reason. One sort of interesting sociological point about this, we originally tried to do this in a way that I thought was much cooler, where people would basically submit code, and then we'd run the code in Docker containers and stuff like that. And I was like, oh yeah, that's totally the right way to do it. That turned out to be hopelessly naive because of basically just the state of the field right now. So for example, again, 99% of neuroscientists that are writing algorithms to solve this problem are using MATLAB. And it turns out putting MATLAB in a Docker container is somewhere between difficult and illegal, depending on who you ask. And I didn't really want to wade into that. So we backed off, and we basically said, all right, people run your algorithms however you want. Just send us the results, and we'll make a leaderboard. And yeah, hopefully this will come to give us some sense of how at least these algorithms compare across different data sets. All right. So I've talked a lot about analysis, but visualization is sort of the other side of analysis. And visualization in science right now, I think, has a lot of the same, we're in sort of the same situation. We have a lot of tools. They're useful. They work. There's a lot of reinvention. There's a lot of development of sort of custom visualizations often that then are not shared or not easily shared across groups. And I think a lot of the same ideas about modularity can be brought to this problem as well. So just very briefly, a couple of open source projects we're working on in this space. A lot of our work on visualization comes from Matt Conlin. Matt's super cool. He's a computational journalist at 538, but he spends half of his time doing that and half of his time doing neuroscience and open science with us. So he's a pretty diverse interests. And Matt's been working on an open source project called Lightning that people here might find interesting. So Lightning is basically two things. It's a way of generating visualizations from data analysis environments, but it's also basically a way of modularizing visualizations themselves. So this project is entirely written in Node, and there's two parts. One is a server, and you can run that server by sort of cloning the repo and installing it. You can also just use this Electron app that we made, which basically just lets you drag and drop it into your applications folder and run it, at least on Mac. And the way this works is it sets up a little server that will generate visualizations and that you can send data to from a bunch of different computing environments. So if you're working in Python or R or whatever, and you want to generate some custom visualization made with web technologies, Lightning just makes it really easy to cross that bridge, which is something that we found when working with web tools for visualization became really difficult because you're constantly having to solve this problem of how to get your data into there from outside. And the other thing it tries to do is, yeah, basically modularize visualization. So in Lightning, every visualization is just an module on NPM. So that means it's self-contained, it's versioned, it has all of its dependencies specified, and it can use whatever it wants. And each module or each visualization can then be taken up by anybody who's running Lightning and use it, or they can just use it standalone in a web page. And what this allows us to do is take, an idea for a custom scientific visualization, write a little module for it, and then anyone who is trying to do or work with the same kind of data can use the same thing. So I thought it was really cool when I was at the Allen Institute recently, and someone there was doing some cool, like 3D rendering of neuron morphologies, and basically found a way to make their thing a little Lightning module, and then other people back at Janelia were able to use essentially the exact same thing to render their neuron morphology data. So it's just a really easy way of starting to share and starting to coordinate these kinds of custom visualizations that we have to do all the time in science a little more effectively and a little more fluidly across groups. Another very recent project is something that I think is incredibly cool. This comes from Mykola Lysenko, who recently started working with us. You might be wondering what a rotating bunny has to do with neuroscience. And I ask myself that question every morning. But let me just briefly explain what's going on here. I think that WebGL and 3D is a really important part of the future of scientific visualization, especially for the complexity of the kinds of things we're rendering, the number of points, number of lines, complex 3D representations of neurons. It's really, or other scientific domains. 3D is really important, and 3D right now is still pretty hard to work with. It's not nearly as straightforward as I think like rendering to canvas or rendering SVG. It requires a lot of knowledge about how sort of GL works in general. And Mykola has been working on an incredibly cool new project called Bri Gl. It's an open source project. You can check it out. It's still sort of in early phases, but it definitely works. You can render stuff like this. And basically, it's a way of interacting with WebGL that's a little more declarative. It's much more straightforward. It's more streamlined. There's less boilerplate, and there's less complexity compared to a lot of existing ways of working with 3D. So we think it's a really exciting way to do 3D in the browser. This is just one of many cool demos. If you go check out the project, you can actually spot Max in this photo, but I'll let you try to figure that out. All right, so that's a lot about analysis and visualization and sort of how we are trying to coordinate a lot of these things across groups. Another really big part of this whole problem is sharing. And to me, sharing means a lot of things. It means being able to have one group run the analysis that another group did. It means being able to disseminate our work in a way that's open and sort of fluid and flexible. And it's something we've been thinking a lot about in the context of neuroscience. And I call this the 1 to 2 problem because I've been really struck by basically being on the ground and watching how science happens, the degree to which we are really limited in our ability to share, even at a very local scale. So I know cases of postdocs in labs where the 2 postdocs are literally taking data from the same instrument, they're doing basically the same experiment or the same kind of experiment, and they're kind of doing the same analysis, but they use completely different tool chains, and they're not even able to share data with each other. And look at each other's data, or like run the analysis that one person ran on the other person's machine. So that's not true throughout science, but I think it's true in a lot of science. And being on the ground, kind of watching that happen, I've been thinking a lot about at that local scale, how do we start solving that problem? So I don't have all the answers, but I want to tell you about one project that came to us really through that process of thinking about how to start doing that. And this project is called Binder. And it's really, all the development is due to Andrew Osheroff, who's an incredibly talented engineer on my team. This is him trying to use a hotel coffee machine. Andrew is here at the conference, so you should definitely talk to him, and he'll be around Berlin for a few days afterwards as well. So go find him afterwards. This project really started with our interest in our use of Jupyter notebooks. I assume a lot of people here know about the Jupyter notebook. Raise your hand. Oh, wow. Okay, great. So almost everybody. Super cool. So I think Jupyter is an awesome way to disseminate at least certain kinds of information or organize certain kinds of information in science. At the very least, it's a way to combine code that you can execute to generate figures and put the whole thing together in a sort of document that can then be run and basically recapitulate the work that you did. And I know there are sort of similar things in some other languages as well. So I love this pattern. And one thing that we tried to do recently for a few projects was to, again, to try to go from this problem of 1 to 2, was to basically take work we were doing for a set of analyses of an experiment, and which often took the form of Jupyter notebooks, and just put it into a GitHub repo with the code and the data, and let that be basically a sort of standalone representation of this particular piece of work. So this shouldn't seem radical at all, but it's actually pretty uncommon in definitely in neuroscience. So here's an example, a GitHub repo. This was actually all the work associated with a paper that we ended up submitting to a journal, but we did this about a year before the paper was even submitted. We just said, we're going to put all the notebooks, we're going to put all the data, and we're just going to put it on GitHub. And that's cool, because then Nick can organize it and track versions, and other people can clone it and play with it. And again, this shouldn't seem particularly radical, but it is actually pretty rare. And one of the things we encountered while doing this is that although GitHub is great for sharing code and sharing, to some degree, sharing data, I'll say more about that in a second, and definitely it's fine for sharing notebooks, it doesn't solve the problem of having this environment, or whatever environment this was run in, now usable on someone else's machine. So there's an extra step to go if someone who is trying to collaborate with Nick, again, even in the same lab, wants to now just be able to rerun exactly the analysis that Nick was using. Or maybe Nick wants to do a lab meeting where everyone in the lab meeting is running the same analysis that Nick was running on his data. How do we do that? That's an additional level of complexity, and often you end up with people installing all kinds of stuff on their computer, and it's complicated. So Andrew and I were thinking about how to start solving this, and we became interested in basically using containerization technologies like Docker to try to make it a little easier to take notebooks and data and code and bundle it together into environments that basically anybody could run, and ideally anybody could run straight from the browser. And we tried doing that a few times for a few particular projects, and found that it was useful and it worked pretty well, but it wasn't streamlined, and we had to sort of redo it and go through it every single time. And then we had a really inspiring conversation with Fernando Perez, creator of IPython and Jupyter, and Fernando basically just said, and I'm paraphrasing here, he pointed out that if you take repositories and notebooks, it can be a really effective way to disseminate scientific information, and possibly also be a torpedo aimed at academic publishing, which I'm all for. I feel at this conference I've seen a lot of torpedoes that can be aimed at academic publishing, so hopefully we're helping with another one. And yeah, the idea here was that if we can make it really, really, really easy to take repositories that have code and data and notebooks and put it into a form that could basically just be deployed straight from the browser really easily, it would be a way to start just more fluidly disseminating the kind of science that we're doing and make it easier for people to reproduce it. So that's what we built in what started as a prototype and is now been used in a lot of different really cool domains. So this project binder, you can go to this website, mybinder.org, and the way this works is basically you just tell us a GitHub repo, and that repo has to have a certain set of contents, and it has to have whatever code and data is necessary to regenerate or rerun your notebooks, and some really minimal specification of the environment. We've been working on not having people have to write, for example, complete Docker files, because Docker in general, I think, is just a really complicated tool for a lot of people to use. So we've been trying to streamline that and take away a lot of the complexity. And then basically you just click this button, and what we do behind the scenes is build for you a Docker image that has all the code and all the data in your repository. And we put it into a form so that you can now embed this little button in your GitHub repo. And when someone clicks on that button, it just immediately launches in the browser right there into the environment in which all this stuff was included, and it's the environment in which all this stuff was originally run. So this now means that Nick at his lab meeting can basically say, hey, everybody in the room, I want you to be doing the analysis that I was just doing. So just click that button, and then everyone in the room is able to do it. So I think this is a cool case of starting where we were just thinking about, again, that problem of like, how do you go from 1 to 2? How do I get Nick to make it really easy to share with the person next to him? And now we're thinking about this and finding that it's being used in a lot of really interesting cases. So we're using it, of course, for sharing code and data and analysis within neuroscience, but it's been used in a lot of other cases. So one is tutorials. This turned out to be a really significant use case. This is just one example. If someone wants to run a tutorial and they want 100 people in the room to all be in exactly the same computing environment doing the exact same thing, Binder's a really easy way to let them do that. And we found it used a lot for that purpose. One that I thought was totally awesome was during the LIGO gravitational waves discovery. So LIGO is super cool. They publish a lot of their data, and they publish a bunch of notebooks. And then Min took their notebooks and put them into a binder, which meant that basically anyone could now just, again, straight from the browser, clicking on a button, could be reproducing these super cool analyses. This was an example that brought down our servers. I woke up at 3 in the morning, and we realized that I didn't even know what had happened. I was like, why are 5,000 people trying to launch what is LIGO? Yeah, and then we realized what it was. I think it's worth pointing out, some people say, well, what's really the value in doing, in sort of being able to reproduce someone else's analysis? Like, yes, you can go in here and change the colors of the lines and stuff like that. I don't know. I don't understand this stuff. I'm not able to really make sense of it. But to me, if there's even one person that is able to rerun this, and it's really easy for them to rerun it, and they either start a collaboration or they find something cool that wasn't there before, like, I already think that's progress. Because again, in a lot of cases, we haven't even made that step of going from 1 to 2. The last one, and this has taken us way outside of neuroscience, which I'll get back to in a second, is it's being used in a lot of really interesting journalism cases. So this was one from BuzzFeed, this guy, Jeremy Singer-Vine, who put together a binder on analysis of US refugee data. And this I thought was awesome. I really believe that any data out there that's relevant for decisions being made by governments, by societies, by people, we should have access to all of it. And if the analysis of those data is relevant to decisions that we or governments are making, then we should be able to analyze it ourselves and see if we agree with the conclusions that are drawn. So as a model for how to disseminate data and visualizations and analysis and topics that are relevant to us, I think this is really important. And it's been really exciting to see it used for this purpose. One thing that Binder doesn't address at all, and has actually been a really big issue, is data sharing. Because I said sort of casually, you can just put a little bit of data in your GitHub repo, and then it'll be included in these environments. But that's not really a stable solution, because what if you now want to be sharing reproducible environments that depend on data that's about a terabyte? Or even data that's not that much, not that big, but big enough to be annoying. So we're really excited about the DAP project, which you heard about yesterday. I think it's a really cool way to think about sharing of all kinds of data, possibly at some point actually sharing of environments themselves, but at least in the short term, sharing of the data that will be a part of or could be a part of these computational environments. So we've been talking with the DAT team about how to integrate these two things and use them together. Any questions about all that stuff? Okay, cool. So at the end here, I'm going to try to get back to brains. And this is basically a story of the life of my lab, which is that although we are neuroscientists, we end up getting involved in a lot of stuff that takes us really far outside of neuroscience. Yeah?
Just to step back.
Yeah.
To the fact that you said, are there any questions?
Great. Oh, cool.
The microscopy imaging and the guy and the complicated, thank you. The microscopy images and the guy and all those screens.
Yeah.
Right? Whatever his name was.
Nick,
And
yeah.
He had a Jupyter notebook.
Yeah.
And it was alive.
That was live, yeah. So we
How
were.
did you do that in a
Oh.
Jupyter notebook?
Oh, you mean like how is the image being rendered live, or
Yeah,
just how?
through a notebook.
Yeah. So in that particular case, I'm pretty sure. Yeah. Basically, the data's coming off the microscope. We send the data from the microscope directly to, in this case, a set of machines or a cluster on campus. There's some crunching that happens, some analysis that happens, and then that data gets basically just absorbed back into what is it, an HTML rendering inside of the notebook. So there's a little lightweight web-based visualization within the notebook that's rendering stuff that's coming off of this analysis that's going from microscope to cluster back to notebook. Yeah. And all the code for that should be on GitHub in some form. All right, so at the end here, I want to try to get back to brains. I know we've taken a tour through a lot of different things that are far from the brain. But that is ultimately both what drives the work that we do. It is always the starting point for all these tools that we try to develop. And it's also what we're ultimately passionate about. So we're going to go back to the mouse on the ball. And I'm just going to give you a little bit of a sense of the kinds of data that we, not only the data we collect, but the kinds of inferences we make and the kinds of things we're trying to learn from the data. So a lot of what we've been doing for the last couple years has been focused on just trying to understand basic relationships between the animal's sensory experience of the world and the way in which neurons in the brain represent that sensory experience. And really that basic goal of establishing a relationship between world and brain has been sort of the bedrock of neuroscience for the last 40 years or so. And in practice, that looks kind of like this. So this is a plot that we'll walk through. What we're looking at here are the responses or the activity of neurons over a period of time where the animal is experiencing the movement of a wall relative to itself. And that's diagrammed at the top where the mouse, you can see with his little whiskers, the wall starts off pretty far away from the mouse, and then the wall starts to move in. So at the beginning it's far away, and then it's here. And then it gets locked at that position for a brief period of time. And what we're seeing in the 4 different colors below are the responses of 4 individual neurons. So neurons do these things, fire these things called action potentials, which are sharp bursts of change in voltage. And each little dot there represents one of these action potentials. We call them spikes. And these are actually based on electrical recordings as opposed to the imaging methods I talked about before. And each of these 4 neurons, we're looking at multiple repeated presentations of that wall as it comes in close to the animal. And the thing to appreciate from this plot is basically just that there is a systematic relationship between how these neurons are firing, how they're responding, and this property of the world, which is how far away is the wall relative to the animal. So in this case, the wall starts off far away. Neuron number 4, the red one, is chattering away. And then as the wall gets closer, now neuron 4 stops chattering, and the other ones start to come on. Neuron 3 is actually suppressed. The yellow one is suppressed in its activity when the wall gets to a certain position. And these are just 4 examples out of many neurons that we can record from. And it just shows you the sort of richness and diversity with which individual neurons in this part of the brain that is encoding information about this sensory experience, the way in which they respond in both space and time to the movement of this aspect in the environment. And then you can also look at it like it's looked at on the right, where we plot the firing rate or the overall activity as a function of the position of the wall and see that these neurons are tuned to the wall position. That is, their response peaks at a particular position of the wall relative to the animal. So this is just looking at a couple neurons at a time. By using these imaging methods that I talked about before, we can basically make maps of this kind of tuning or this kind of response properties across really large fractions of the brain. So again, here's a movie where we're seeing in each of these little bright flashes an individual neuron, the activity of an individual neuron, and when they sort of pop and the light goes quickly on, when it sort of brights up and then comes down, that's an action potential. And we can make maps by basically coloring every pixel in this movie based on the relationship between the response of that pixel, or the fluorescence signal that we see from that pixel, and again, some property of the world. So in this particular map, which I just think looks super cool, we are coloring every pixel. The blue ones, the blue pixels get colored when their response is well predicted by the position of the wall. So that's the property we were just looking at. The orange ones are actually predicted well by the running speed of the animal, so how quickly she was running on the ball. And then the green ones are pixels that respond reliably, but we don't, or at least are not able to directly relate it either to the wall position or the running speed. So these kind of maps give you a sense over, potentially hundreds or even thousands of neurons, give you a sense of how large collections of neurons in the brain are encoding different properties of the outside world. And we can zoom in. The level of detail is phenomenal. Again, this kind of picture is basically what we had access to before for a long time. And with this new microscope, we're able just to look at a much larger fraction of this whole space simultaneously. And I think it really changes the picture in terms of how you now think about how different parts of the brain are representing different aspects of the world. So Nick and I have spent a lot of time thinking about, again, encoding of these basic sensory variables. How does the mouse basically know where the walls are and where it is relative to the walls? But we also want to think about more complicated behaviors, and we're working on how to do that. And studying more complicated behaviors, by that I mean things like learning and decision-making and memory. How does not only the mouse know sort of where it is relative to its environment, but how does it know where to go next, and how does it know where it just was, and how does it plan, and how does it make decisions? So in the virtual environment we've had so far, where there are just walls on either side and the animals in this moving corridor, it's been hard to study that, because if all you have is a corridor, you can't do that many complicated things. But we've recently been trying to build, and have recently managed to build a new version of this setup that lets us ask sort of another order of complicated question. So this is basically the same thing I was showing before, but now there are 3 walls instead of 2. It's kind of crazy. So the way this works, there are walls on either side of the animal, and the walls are moving back and forth. And then there's another wall in the front, and that wall actually has 6 degrees of freedom, so it can move forward and back, it can move left and right, and it can rotate. There's a little servo motor on it. And basically, all these little walls do this ballet around the mouse that makes it think it's inside of an even more complex virtual maze. And that's shown on the right, where we're basically looking at a top-down representation of this virtual maze, and the white circle is the position of the mouse in this environment. Again, the mouse isn't really, she's running on the ball, but she's not moving anywhere. But because of all these walls, she sort of thinks she is, or so we think. And, what exactly, what exactly the motors do to create this illusion is maybe not obvious just by watching this video. The intuition is that basically, if you're a mouse and you're approaching what is going to be a choice point, so a point at which you can go left or right, as you walk up to it, we have these walls on the left and right. They start to move away, and then the wall that's going to represent the choice point starts to come in towards you. And then as she thinks tries to go a little bit to the left or a little bit to the right, that wall rotates a little bit to the left or right. And then as she commits to one decision or the other, this one sort of rotates and then moves out of the way, and the other wall comes in from the side and replaces it. So Nick is some kind of savant at coding motors. I don't know how exactly he figured out how to program all these things. It's really remarkable. And we have some basic evidence that this kind of thing is at least capable of creating for the mouse an experience of maybe getting some sense of its environment. We're still in the early days of prototyping this. I want to stress that. This is very anecdotal. But it's just one example of the kind of behavior that we think we might see, or we at least hope to see, look something like this. So here's a case where we're looking at traces that represent the behavior, again, the position of the mouse in this virtual environment on a sequence of trials. And the first time, or in this particular case, I should say, the white square is a location that if the mouse gets to it, she gets rewarded with a little drop of water or juice from the lick port. On the first trial, she gets kind of confused and goes left and then goes right and gets confused again. Gets really confused on the next 2. But then there's another set of trials, 4, 5, and 6, where she basically nails it every single time. It gets straight to that location. So again, this is super anecdotal, but in principle, if we can elicit this kind of behavior, and in particular this form of learning something about the structure of environment based on very little information, this is something we're really interested in, because basically, if we can figure out what's going on in the brain by watching all of it, which we can now do, between trial 3 and the trials that come after it, I think there's something really powerful there. If you look at a lot of work in things like artificial intelligence now and neural networks, learning from small amounts of data is a really hard problem. These models and these really complex systems have been really good at solving a lot of complicated problems when they can learn from enormous amounts of data. When big companies are scraping all of your data to learn all these really complicated models, they have an enormous amount of information. But I think organisms and animals, definitely humans, we can learn with a very small amount of information. And understanding how the brain does that, I think is super interesting. And it could be something that's kind of unique about the computational complexity of this thing. So it's something we're interested in. One thing about those motors that, again, brings us back, we sort of can't stay away from technology, that brings us back a little bit is the idea of taking some of the modularity ideas I talked about before in the context of analysis and actually bringing that to hardware. So this is kind of a crazy thing we've been playing around with recently. This is, again, Nick. I wonder if he knows that I show all these videos. So we do a lot of our work out of coffee shops. So this is Nick at Pete's. We have this box of hardware now that we bring around when we go to the coffee shop. And basically what we've been playing around with, if you go back to that picture of this setup, this is a really complicated experiment. And right now, Nick is basically programmed this thing as a monolithic pile of C code that powers a real-time Linux machine, which, it works, and he got it to work. But if any other human was to try to do exactly this experiment, it would basically be impossible. And that's even true within individual labs. People basically program these monolithic piles of code to program experiments that involve basically moving stuff around and rewarding the animal when it does things. And it's sort of, it's not unlike the analysis problem in that different groups do things differently, but there are commonalities and there are similarities. So we've been really interested in whether we can basically use a combination of Node and JavaScript and Electron and a lot of ways of interfacing with hardware, in particular these new really cheap microcontrollers like Arduinos and Raspberry Pis, to do or program these kinds of experiments in terms of modules that are much easier for us to share and coordinate and collaborate on across groups. So what's actually going on here is Nick is playing a game. It's not as complicated as the full 3D virtual environment game, but he's playing a game that's actually not unlike the tasks that a lot of labs have mice do, except instead of being powered by this pile of C code and a real-time Linux machine, he's basically running it in Node. So that little window there is an Electron app. And Electron's really cool because it lets us combine web views with runtimes that integrate with things like hardware. So talking to hardware from your browser is kind of complicated, but in Electron it's super easy. And he's playing this game where there's a little bit of rendering really lightweight UI right now that's rendering something related to the state of the game. But you can imagine that would be the motors, or that's something about what's being delivered to the animal. And then he's playing this game and getting really frustrated because it's a really hard game. And we're just getting started with this, but I think it's kind of a cool way, or it's a cool way in which technology is maybe going to allow us to start modularizing and making it easier to share and collaborate on not only the analysis of the data, but actually the way we do the experiments. And I just think it's a really cool world if we can actually start talking about experiments as basically collections of modules, and some of us are doing the same experiment, some of us are doing different experiments, and we can start sharing and coordinating that a little bit better. Partly driven by doing this stuff, and this is the final thing I'll tell you about, we got interested in whether we could try to connect some of the stuff we were doing in the mouse and some of these virtual exploration experiments with behaviors that we could study in humans. And there's actually a long history of this. It's something I did. I used to study the visual system, and it was always really interesting to me to think about the relationship between how humans perceptually experience the world and what we can study in animals. So in animals, we have a lot of limits in terms of the behaviors that we can ask animals to do. We can't talk to them, so it's hard to understand exactly how they're experiencing things. And it's hard to get intuition about how they're experiencing things, and it's really low throughput. We can only run 1 or 2 mice a day. When you're working with humans and you're doing experiments with humans, in the good way, like you're measuring behavior, you can do it really high throughput because we can use the web to disseminate behavioral experiments. And you can introspect. You can ask yourself, how am I experiencing this behavior? How am I experiencing this image, or how am I experiencing this task? So it was basically the combination of that concept and the fact that we were starting to program these tasks in JavaScript that we decided to try to build basically a game that could be run in the web browser that would be a sort of analog to the game, essentially, that we were having the mice play in these experiments. So obviously it's built out of hexagons. And this is a game called Hexaworld. Here, hexagons actually really make sense because when you make those choice points in the mouse maze and you chain together choice points of that form, you basically get hexagons naturally. So the way Hexaworld works is there's basically a maze-like environment. Think like Pac-Man, but on hexagonal grid. For humans, of course, we can't stimulate the whiskers. Not all of us even have whiskers. But we can use visual information. So this example, there's a maze. You're at one location in the maze. This is like the top-down view. And then the colored squares represent locations that are going to act as visual landmarks when you're inside this environment. We have a little web-based thing to let us design these worlds so we can build little environments. And this is kind of cool because now we basically can use the exact same thing to build the environments that we make for the human version. We can use the same thing to build environments that we use for the mouse version. So that's been kind of fun. Again, this is all very much in progress. And then what it looks like, this is a very early version rendered in WebGL running in the browser. So basically that little white character is you, and you're inside of this maze, and there are walls, which again correspond to the walls that the mouse was experiencing, and there are these visual landmarks off in the distance. And we're still just getting started and thinking about what are like cool tasks or sort of game-like behaviors that we can get people to play. Turns out there's a really interesting challenge in how do you make something that's fun for people to play and something that people actually want to play, but also lets you answer the scientific questions we're interested in. You know, here we want to basically have hundreds of people play games like this and then use their behavior to understand something about behavior in general, and in particular behavior that's going to inform the kind of experiments we do in animals, where we can actually look at the activity of brains while animals do these kinds of tasks. So we're just getting started with this, but it's been a really fun way to think about, the differences in relationships between animal experiments and human experiments, and also the kinds of things we can do kind of large-scale behavior in the browser. I should say all the code for this is already on GitHub, though it's like super early, but if anyone wants to check it out. And any data that we collect as part of this sort of behavioral experiment, we will make available. So it's really important to us that we will do that. So with that, I'm totally happy to take questions. Thanks so much for listening. I want to thank all the people in my group. We're a really diverse team. We basically all work on some combination of all of these things, and it's been an amazing group to work with. I'm really lucky to work with all of them. We maintain a couple chat rooms, so we have a Freeman Lab chat room, which is a public chat room for our lab. There's a lot of crazy stuff in there basically every day, whether it's like pictures of brains or weird 3D visualizations or all kinds of crazy stuff, so you should come join that. And then also, Max and I started this Code for Science group, and that's a great place to talk about all kinds of things related to computation and coding and modularity and all that fun stuff in the context of science. So thanks very much.
So that was awesome and really inspiring. I don't work at anything like this, and so it's great for the conference to get exposed to this kind of thing. What do you do about describing the data? So if I get a lump of data from somebody, I need to know where it comes from, when it was measured, how it was measured, degree of accuracy, all this kind of stuff, and that also has to be something you have to coordinate with other labs as well. So what do you do about that part?
That's a great question. I'd love to tell you the state of it in neuroscience right now, which is that basically there's almost no coordination of metadata or information about data sets. So I can tell you, we're trying to find, I would say right now, just very contained spaces where we can even just prototype how to solve that problem. I think, again, this thing about, ideally we'd have a way to standardize the formats that hundreds of labs use to represent this kind of data. But I don't even know how to get two postdocs in the same lab to like standardize how they represent their data. So I'm trying to solve that problem. We've been doing it where we can, in a few cases, if there's like a particular experiment that people are doing, even just try to get, say, 4 people in the same lab to agree, and almost use that as a little prototype or experiment to figure out what is the kind of metadata they want. My sense in general from having watched a few projects that have tried to do this is that, this is just a running theme, you want to solve these things in a modular way. I think it's really, really hard, given the rate at which things are changing in the field, to come up with something that's even going to be a good description for, say, all of, definitely not all of neuroscience, even like all of mouse neuroscience is really hard. I think something like all of 2-photon imaging maybe is a little more tractable. So we've been trying to figure out how narrow can we make the scope, and then sort of prototype solutions. At the end of the day, it will have to mean something like labs that are collecting data need to encode some kind of metadata about what the experiment they did and make it really easy for them to do that. But I think it's about finding the right scope and getting it to work in actual labs on the ground.
Hi. I'm from the neuroscience field myself,
Okay.
and one problem I always had when using your amazing tools like Funder is that actually it was very hard for me to, sometimes there's no money for Amazon Web Services and the scientific grants, right? And while it's easy for me to get hands on supercomputers, it's very hard to get the admins running Spark on it, right?
Yeah.
So there was kind of thing that actually limited a bit the usefulness of Funder, and so I don't know if you have some advice how to get money for AWS or.
Yeah. Well, I'd love to just, it's a fantastic question. It's something we've thought a lot about. One thing we've been doing recently in the context of these particular tools has been trying to work on basically making it a lot easier to not necessarily rely on things like Spark. So we spent a lot of time thinking about using Spark for distributed computing to make a lot of these workflows way more efficient. But one thing we realized is that partly for this reason, there are a lot of cases where that's just kind of impractical. And most things, not all things, but most things will run locally. It will just take longer. So something we've been working on with these projects, very recently we released all this stuff, is to basically have uniform APIs that work identically across local and distributed settings. So everything as part of Thunder now has this property where basically you can just install it as a local Python package and everything will work and there's no dependency on Spark. If you are on a cluster, then the exact same code and the exact same APIs will now run in a distributed fashion and will hopefully be much faster. But if you're local and you're just doing stuff locally, then everything will run as well. It will just take longer. So basically one answer to your question is just to not rely on Spark because it's a DevOps problem. It's a complicated problem getting these distributed computing tools set up. It's hard within a local institutional cluster because you need to rely on local support, and it's expensive if you want to use cloud compute. So I'm not super familiar with, I know there's some recent pushes, I think at University of Washington was one example I heard about, to try to change some of the discussion around the way grant money gets paid for cloud compute support. I've heard that at least in some universities, there are weird loopholes that make it way more, it's cheaper to put the same money towards local institutional cluster than towards cloud compute, even if that's not the rational decision for the lab to make, because of basically just weird loopholes in the university where they want to be building their own infrastructure. So that sounds crazy to me, and hopefully I think some other people feel that way too and are trying to make it easier to get grant money to pay for AWS. But I don't know sort of across the board how that's going. But yeah, I sympathize. No, I mean, honestly, if we didn't, I would love to never have to deal with distributed computing. And we've been trying to make it at least easier to do that in some cases, but sometimes it is kind of at least super useful if not necessary. Yeah.
All right, I think, one last question. We can do, well, you already had a question, so I'll give you the.
I'll be around.
So I just wanted to ask about the imaging again. So you mentioned that recently you managed to start imaging a larger area of the brain.
Yeah.
But it's still not the whole thing, right?
Definitely not, yeah.
How far do you think you are in time from being able to image the whole brain using something?
Entire mouse brain. That's a great question. We talk about it a lot. I think the entire mouse brain is pretty far away. To put it in sense of perspective, the mouse brain is maybe about 80 million neurons. Those images I was showing, the best possible thing we can do right now with this new mesoscope is about 10,000 neurons. So we are looking at a small fraction of the whole brain. There are other animals. So I've done some work in the larval Drosophila, the fly, and also the larval zebrafish, where it's possible, basically because the zebrafish is transparent. So it turns out to be way easier there are microscopes that you can look at the entire, basically every neuron in the brain at once. It also has way fewer neurons, there's 100,000. There are variants of the methods that I was showing here that could get us closer, larger fields of view. So right now I said we have this window that's maybe about 5 millimeters, which for us would be about this. You can make that larger, but having access to all depths at the temporal resolution required to resolve the timing of individual action potentials over the entire brain. I don't know, 10 years? And then we'll talk again. I don't know, but it would be amazing. It's probably going to take a while, and probably will take some really big new ideas. That's my guess.
All right, so that's all the time we have for questions, but I want to say thanks for sharing all that awesome work, and thanks for coming. Thanks.